All right. So remember my simplified version of glycolysis. My favorite part of the metabolism lecture is I kind of imagine, Dr. D, who is very much alive, but if he were not like rolling over in his grave as I, simplify biochemistry down to Dr. Daft style. All right. But right, I think, sometimes we get lost in the details of the metabolic pathways and forget some of the small things, right? So glucose is 6-carbon sugar, and you're ultimately going down to pyruvate, and we already talked about. You have 2-pyruvate because what is biology not, Jenna? Magic, right? I would have pulled a coin out from behind your ear, but I don't know that trick and social distancing, so, I'm not. I'm also not saying that we don't appreciate glycolysis, but for this class, I'm certainly not interested in getting bogged down in necessarily every metabolite that's produced, along with every enzyme, right, that is driving that reaction. Okay. But I do want you to appreciate, right, that there is investment, right? The biggest reward that we're reaping is the generation of this reducing power, right? And then we're setting up, right, these pyruvates. Now, pyruvate is going to be really vital because pyruvate, Caitlin, what's the next step after I make pyruvate? Where do I go in the central metabolic pathway after that? I'm kind of like, thank you for not even toying with my emotions there, but I think you do know. What comes after pyruvate gets converted to acetyl-CoA. Where does acetyl-CoA go? Citric acid cycle? Who said that? Whoa, good, right? TCA cycle, citric acid cycle, Krebs cycle, right? What we will also talk about maybe on Friday is that pyruvate is going to actually be important as a terminal electron acceptor when fermentation occurs. All right, so those of you, right, we'll talk about this fermentation. What's some of the wonderful things that fermentation produces? Yes, cheese, delicious. What's your favorite cheese?
I like the mixed, like Mexican cheese.
Okay. Like the shredded? Okay. I'm a sharp cheddar smoked Gouda type of guy, but I still love that cheese too. I mean, there's not many cheeses that I say no to. All right, cool. So cheese, what else? What else do we get from fermentation? Huh? Got you. You are busted. All right. It's true, right? Rubbing alcohol, right, for wounds, right? No, exactly, right? So beer, wine, right? They're made from fermentation. What else? What else comes from fermentation? Lactic acid does come from fermentation. That's true. I think more food products. Well, yeast doesn't come from fermentation. Okay, yes, yeast ferments, right? So breads. Yeah, there we go. It's like, right, breads. Ooh, cured meats, salami, delicious. Ooh, hot sauce, also delicious, right? Are all, right, because of pyruvate. Pyruvate is getting converted to things like Jennifer mentioned, lactic acid, right? And so you're gonna see how that plays out here in just a little bit. Glycolysis, again, we don't spend a lot of time on here, but as I just mentioned, I want you to appreciate that there is an investment phase. And so you are taking ATP, right, and popping it on to glucose, right, and fructose 1,6-phosphate, right? So you spend 2 ATP, right? You generate 4, so that's why you have your net of 2. Again, really important here, the NADH, right? And this is the same principle in eukaryotic cells and prokaryotic cells, right? So we love reducing power. Holmes, if I ask what glycolysis is for, and you say generating ATP without saying through reducing power that's sent to the electron transport chain, right here. All right, and I don't like crying. So let's talk about the pentose phosphate pathway. All right, and the reason I have this next is because you'll see there's an important interplay between this pathway and glycolysis. All right, so glucose 6-phosphate, right? So glucose has entered the cell, it gets phosphorylated. When glucose gets phosphorylated, it has two options, right? It can either go into the pentose phosphate pathway or it can continue down the pathway of glycolysis. Does anyone remember from last week what would drive this decision? How would I choose which pathway to go down? It's dependent on what? What you have or what you need, right? Perfect, I can take that, right? So the energy requirement of the cell, if it has energy, well, then you can go down the pentose phosphate pathway, because we're going to see here in a second, the pentose phosphate pathway is very anabolic in nature, right? Things get built from it. If I don't have energy, though, then I'm going to go down glycolysis to continue breaking down those carbon molecules to generate a little bit of ATP and to generate that reducing power that will hopefully go to the electron transport chain, right? So here's where we see NADPH, all right? NADH is present in glycolysis, and you'll see here in just a second the TCA cycle. FADH is also found in the TCA cycle. The only place that we find NADPH, right, is going to be in the pentose phosphate pathway, all right? Two of the very important metabolic precursors that are produced from the pentose phosphate pathway are ribose 5-phosphate and uracilose 4-phosphate, right? These are two essential building components for amino acids and for nucleic acids. Okay. Yeah. That's important. Got it, Rachel? Okay, good. So again, we already mentioned kind of an alternative to glycolysis, right? You guys just told me energy dependency of the cell, all right? And I mentioned nucleic acid and amino acid can also be used for fatty acid synthesis. So this pathway, this pentose phosphate pathway is essential, right, for those macromolecules that you know and love. Glyceraldehyde 3-phosphate, G3P, fructose 6-phosphate, right, are also molecules that can be shuttled back and forth between glycolysis and the pentose phosphate pathway, okay? So again, maybe you made a commitment, you thought you had enough energy, something happens to the cell, it realizes it doesn't have enough energy, but that's not that big a deal because there's multiple points within glycolysis that I can shuttle, right, molecules to the pentose phosphate pathway or vice versa, right? So there's a lot of interplay. So if you'd go back a few slides to that original diagram, right, you saw that the pentose phosphate pathway is over here, glycolysis was over here, and the TCA was over here, right? Because these two, right, can share and can go back and forth. Now there's two phases to the pentose phosphate pathway. There's the non-oxidative, and this is where we're generating NADPH. This is going to be reducing power that we use to drive anabolic reactions, okay? So NADPH is not making the journey to the electron transport chain, right? It's going to be used immediately, right, to drive these anabolic reactions. And then the non-oxidative, right, is where you're generating these precursor metabolites that will allow you to then build these different macromolecules. Okay. Now, I'm not going to ask you to draw these, although I think, Morgan, you could because of biochem, right? Maybe. Ooh, game on. All right. So Morgan, I'll have a special question on your test to draw D-glucano 1,5-lactone 6-phosphate. All right. But I just, wanted to show you, right, the generation of NADPH. Here is glucose 6-phosphate. All right. So again, kind of that important, hey, which way is it going? Molecule. So one molecule of NADPH, another molecule of NADPH. All right. Again, all right, this is dependent on NADP being produced. If you don't have NADP present, well, then you can't generate NADPH. That electron and proton, right, have nowhere to go. All right. Yeah, don't care about that. This is the non-oxidative pathway. Again, I'm not going to ask you to draw this out, but again, here you see glucose 6-phosphate. Farther down, right, here's your glyceraldehyde 3-phosphate, and here's your fructose 6-phosphate. So you can still bring those, right, into this pathway or generate these in this pathway and then use them, all right, to go back to glycolysis if needed, right? But here's your 4-carbon sugar, here's your 5-carbon sugar that you can then use as building blocks for your other macromolecules. All right. Now, what do we need to know about the pentose phosphate pathway? We're not spending a lot of time on it, but only source of NADPH, right? It's for powering anabolic reactions, all right? You have glucose 6-phosphate, all right? But don't forget, right, that you can also, right, take glyceraldehyde 3-phosphate and fructose 6-phosphate and shuttle them back into glycolysis if needed, right? And then use them to immediately go into the TCA cycle, right? You can convert them to a pyruvate molecule if needed, right? 5-carbon sugars, and I could also say 4-carbon sugars, right? Needed for assembly of macromolecules. What we didn't really spend a lot of time on too is one of the precursor molecules that's generated in the pentose phosphate pathway can also, right, be a molecule that's part of the Calvin cycle. Taylor? So, so I was going to ask you a question, and then you can ask your question. Are you capable of performing the Calvin cycle in your cells? Good. All right. Proceed with your question.
So if the 3 and 6 carbon molecules go into glycolysis in the TCA cycle, where does NADPH go?
NADPH stays within the pentose phosphate pathway, okay. And if you're not shuttling back to glycolysis, then NADPH is being used to essentially power the anabolic reactions that are building those macromolecules like proteins, lipids, and nucleic acids.
So it doesn't go to like the next cycle?
No. I mean, yes, I would say that it, you know, again, I'm not the biochemist, but I would say that it stays within the pentose phosphate pathway to help drive the anabolic reactions, if that's the way you're going. Okay, good question. And Taylor was right. No Calvin cycle in your cells. Rachel, can you remind me what the Calvin cycle is for? Okay, yeah, yeah. Calvin cycle, plants, photosynthesis. Okay. We're on the right path. I like it. Sarah, help me out. What's the Calvin cycle for? And we had photosynthesis and plants as important clue keywords from Rachel. It takes energy. Holmes, you got me? To make glucose. Okay, yes, to make glucose. From what? I mean, you could make glucose, right? I could put a bunch of organic carbon in you and you could reassemble it to make glucose. So what's the Calvin cycle doing? What is it taking to make glucose? It's CO2. There we go. Oh no, wait a minute. We would not be good at charades together. I think that's what I'm looking at, right? CO2, right? So the Calvin cycle is taking inorganic carbon and CO2 and capturing it and turning it into organic carbon. Now, you guys are all right with light and photosynthesis, right? Because what would you guess, Michaela, the energy investment is to take carbon from the air and turn it into organic carbon that your cells can use? Would that take a lot or a little? It would take a lot of energy, right? The sun, right? Sunlight is a very, very plentiful source of energy. So organisms that are photosynthetic, right, can generate a lot of energy. It takes a lot of energy to fix carbon, so boom, right? And we'll see, and we've already mentioned that some prokaryotic cells are capable of fixing carbon, right? So we have photosynthetic bacteria, All right, so they're going to actually use some of the precursor metabolites from the pentose phosphate pathway as part of the Calvin cycle, which, as Taylor so duly noted, the answer is no, she cannot. Good. All right. Ooh, transition step. This is interesting. You know, I don't know. I assume this is how it's taught, and maybe I just wasn't paying attention in my chemistry classes when I was an undergrad/graduate student. But you know, I always kind of lumped the transition step in with the TCA cycle, right? A lot of textbooks don't do that, so I'll be honest, I'm not going back and leafing through my old chemistry textbooks to see if it was them or if it was me. We'll just assume that it was me, right? So technically, it's a transition step. But you know, this transition step, if it's completed, always leads to the TCA cycle. So in this case, right, this transition step, you're taking pyruvate. All right, you are getting rid of carbon, and you now have acetyl-CoA. Now this CoA is added, right, coenzyme A, and this is going to be the starting product of the TCA cycle. And you see here, even in this transition step, you're generating reducing power in the form of NADH. Yes, ma'am. Correct. Yeah. So yes, I haven't gone back, and I guess maybe I could do that to make it, or maybe I should put, like, I could just put ×2 down here. That would make more sense, right? So yes, so again, you know, Abby's right, because biology is not, what is it? Not, Jenna? Yes, it's not magic, right? And so yes, for every molecule of glucose, you have 2 molecules of pyruvate. For every molecule of pyruvate, you have an NADH. So for every molecule of glucose, you have 2 NADH. See, if you say it really fast, it makes you sound really smart, but I was just doing 3 + 3 is 6. That's all I was doing there. So yes, that is correct, right? That's where I'm getting the 2 NADH. I'm also not a math whiz, but yeah, I assure you in this case that I'm not that off, right? Right, Alanis? All right. All right, so this is coenzyme A. All right, this is coenzyme A. I think we really need to appreciate its addition, right, to the acetyl group, because the addition of coenzyme A is going to allow, right, this molecule, this acetyl-CoA, to interact with enzymes that are going to drive the citric acid cycle. Okay. Again, unless your name is Morgan, I will not require you to draw this out on an exam. What's going to be really challenging, Morgan, too, is I have no idea how you would draw this out on a Moodle exam, but I'll leave that up to you to figure out. Okay, good. So again, kind of going back, you know, to Abby's point, right? For every glucose, right, there's 2 cycles because you are generating 2 molecules of acetyl-CoA because you had 2 pyruvates. All right, and you're going to generate, you'll see here actually in just a second, a large amount of NADH and also FADH2. So here's the TCA cycle. Again, I'm not going to ask you to draw this out or memorize it, but I again want to point out a couple things. Look, that's all the ATP that you're generating directly right there. 1 molecule of ATP, right? So for every molecule of glucose, you're only getting 2 molecules of ATP. I repeat, Hannah, don't let me down, right? If I ask the purpose, right, or the function, and you just say to generate ATP without indicating to me that it's the generation of ATP through the production of reducing, right? I want you to be specific there, because again, the pathways themselves, there's very little direct ATP production, right? A couple other things, right? Oh, do you guys remember seeing this in lab? Citrate? Do you remember doing that? You don't remember doing that? You do? Abby, I just don't know what to believe anymore from you. What was this test? The citrate slant, right? And the citrate slant was looking at the ability, right, of a bacteria to utilize citrate as its carbon energy source, right? What color did it turn if the bacteria was able to break down citrate and utilize it for energy? Remember? Man, I wish Professor Perry was here. The rainbow, one of those colors, right? No. So it was green, and it turned blue. All right. Yikes! All right. That's okay. You guys are on your unknowns right now, right? I'm sure you've gone through a lot of tests, so it's all kind of getting mixed up. Yeah. So you've done the citrate slant, right? So that's what that is. That's what that's showing. There are a lot of things that turn yellow, though. So who said yellow? Okay, good. So there are a lot of things that turn yellow. I don't think there's any tests that turn green. So I'm a little worried with one of you two back there, but that's okay, right? So that's what you guys are looking at with the citrate test, the ability to utilize that molecule for an energy source. You also, I guess I didn't highlight them. You can see here, like NADH, NADH, NADH. Right, that's a lot of NADH that you're generating right there. And then you also have, right, don't forget about my boy FADH2, also going to the electron transport chain. Can any of my biochemistry nerds tell me why there's more ATP generated per molecule of NADH versus FADH2? That. Man, I've been waiting all semester. The things that I use my brain power for are probably not those. All right. So why? Right. Again, I can't remember. I don't know all of your chemistry backgrounds, but why do you generate more ATP per molecule of NADH compared to FADH2? And they're both going to the electron transport chain, but I'm getting more energy from NADH.
NADH goes to complex 1, and FADH goes to complex 2.
And why? Why does that make a difference?
Because it was 4 ATP.
Yeah, there we go, right? Complex 1 is a proton pump, all right? The whole point of the electron transport chain is to generate a proton gradient. Complex 2 is just kind of more of a, let's hang out until we go to complex 3, which is then also a proton pump, right? So you're pumping out more protons because NADH is dropping off a complex earlier than FADH2. And the first complex that FADH2 drops off at, right, is not even a proton pump, right? So that's why that happens. Okay. And I just think it's kind of interesting. And you'll see here in just a second, yeah, I think we'll definitely get to it, that the complexes in the electron transport chain are different, right? We have a different number of complexes, very similar functions, but a different number of complexes between prokaryotic cells and eukaryotic cells. All right. So again, ATP, 6 NADH. Don't forget too that we're generating 2 NADH from the transition step. And so if you kind of group the transition step and the TCA cycle together, you're actually getting 6 NADH. You get 2 FADH2 plus some other precursor metabolites. And again, remember, this is all, right, because you're going through 2 turns, right? 1 molecule of glucose, 2 molecules of pyruvate, 2 molecules of pyruvate, 2 molecules of acetyl-CoA, right? Again, this is just part of your PowerPoint presentation. I'm not going to read through it. This is everything we just talked about. Again, I think it's wise, but you guys have good approaches that work for you to start with a table, look through that table, and then things that you're not clear of, then kind of delve out. All right, it just kind of puts it all there in one place for you, and then you can kind of move on from there and focus on different aspects of that table. All right, the electron transport chain. All right, this is what happens when you take away oxygen, okay, for us. Right, so oxidative phosphorylation, right, is referring to 2 processes, right? The electron transport chain and ATP synthesis, right? And these are linked together. Oh, this PowerPoint is an excellent question. I'm treating the PowerPoint like as a person. What is that? What's the word for when you give something that's not alive animal-like properties? I think so. That sounds right. Anthropomorphism. There we go. Whew, that's a tongue twister. Boom. That was like one of my favorite things that I learned in English in high school. You know, the rest of it, I don't know. But anyway, right? So, Alanis. Answer this question for me. I mean, cytoplasm is part of the word. So there's complexes just floating in the cytoplasm for electron transport. Let's take a step back here. That's okay. Start here for me. Where does the electron transport take place in eukaryotic cells? Do I need to go reevaluate your schedule? She's like, no, please. All right. Someone help her out. Mitochondria. Yes. Oh, man. Yes. I would say it's Monday, but it's not Monday. It's definitely Wednesday, right? Maybe it's Monday disguised as a Wednesday. All right. So good. Mitochondria. All right. I want to go back to Atlantis, but I don't do it. Thank you for your honesty. All right. Prokaryotic cells. Where? Oh my goodness. All right. Where is the electron transport chain taking place in prokaryotic cells? Please, someone restore my faith in Wednesday. Cell membrane. Okay. That's why Atlantis, I looked at it because you're like cytoplasm, and if you would have said cytoplasmic membrane, it would have been like bing, bing, bing. Right. So cytoplasmic membrane, right? So we have two different locations there. So that's going to be an important difference. And now we're going to talk about some of these complex differences within them too, right? This is my favorite diagram of the electron transport chain, right? This is how my mind, thinks. I like to picture it as a slinky going down the stairs, right? Each one of these steps is a different complex, right? As that electron moves down those steps, right, protons, right, are moving to help establish that proton gradient. So the farther you move down the steps, the more protons you've been able to move. The more protons you have, right, the stronger the concentration gradient, the more energy you can make. All right. Ooh, and then we also have, right, oxygen down here, right? So oxygen's that terminal electron acceptor. You take away oxygen from the system in you toast, all right? We'll actually talk about some prokaryotic cells can be just fine, right, when you take away oxygen. I don't know why I've kept this slide. I got this one from Dr. Chrisman originally, and I'm like, how can you not smile, right, when you see Tibetan monks going down a roller coaster? You just can't, right? Please remember too, if I come back here, and again, this is, the diagram weakness, but I just mentioned it, right? As your electrons move, right, protons are moving too. I think that's kind of one of the things that I wish was emphasized more. I think I told you guys that when I was a student, is like, hey, the protons are moving with it, right? Electrons don't travel alone. Now, right, we're not going to spend a ton of time here. There are a few differences, but nothing major here. Right, you have your large complexes that serve as proton pumps, right? Most of them, you do have complex 2 or a complex 2 equivalency in prokaryotic cells that does not pump protons. And then you have smaller compounds that are moving electrons between the larger complexes, all right? So if you think of the larger complexes as kind of important landmarks, and think about these small carriers as a Uber or a Lyft, right? That's taking you in between the complexes. I know, I'm pretty hip and down with society. I used to say taxis, but I don't even know if you guys know what those are anymore, right? Which is very interesting. Have you ever seen the taxis driving around Cleveland?
No.
There's a couple of them. There's a small company. Yeah, they're like the box-looking cars. And I was like, man, who moved to Cleveland? And I was like, you know what Cleveland needs? Taxis. But I'll give them this. They have the market cornered, so, So there are three types of electron carriers, right? So these are the Uber or the Lyfts that are moving them between the two complexes, quinone, cytochromes, and flavoproteins. Yeah, that's fun to say. Now, For quinones, you have different varieties. So you have ubiquinone, which you find in your cells, an important electron carrier, right? You also have ubiquinone in some prokaryotic cells. Some prokaryotic cells also possess menaquinone, right? And menaquinone is, right, part of the electron transport chain for prokaryotic cells. It also is an important source of vitamin K, all right? And vitamin K, right, is important for blood coagulation. What's blood coagulation? What does that mean again? Clotting, right? So, right, when Morgan gets cut, right? Oh, good, thank you. Yeah. She doesn't bleed out, right? You want your blood to clot. Now, those of you that have taken A&P2, right, you realize that the blood clotting mechanism is pretty complicated, right? But it turns out, like, and at this point, if you don't love your gut bacteria, you're just wrong. But now you can also thank them for one more valuable thing that they do, right? Vitamin K is important for coagulation, and you're getting that from your gut bacteria from menaquinone. Right now, you don't have menaquinone, right? It's a type of ubiquinone, so it's carrying electrons, right? But it's just a different molecule that's present in them that we don't have, right? Not surprisingly, all right, you're going to see these electron transport molecules are lipid soluble, and that makes sense because you're either moving through the cytoplasmic membrane or you're moving through the mitochondrial membrane, right, as you move from complex to complex. So they have to be lipid soluble. Oh yeah, cytochromes, right? These contain a heme group. What other molecule, Alyssa, that you love so much contains heme? Well, iron. So heme contains iron, right? There's something else that you love that contains heme, which does contain iron. Don't overthink it. It's in the name. Mondays. Hemoglobin. You've heard of that, right? Okay, good. Did you take A&P 1 or 2? Okay, then I don't blame you quite as much. Who took A&P 2? Did you raise your hand? You're in it. So you've had the cardiovascular blood unit already. Okay, see. So right, hemoglobin, right? Hemoglobin contains iron, right? Iron's an important part of hemoglobin. What does hemoglobin do again? Carries oxygen. Yeah. What does hemoglobin love even more than oxygen? Yeah, carbon monoxide, right? So if you heard of carbon monoxide poisoning, right, that's what happens to those folks, right? It has a higher affinity for heme, right, than oxygen, and it doesn't like to let go, and so basically you reduce your ability to carry oxygen throughout your body, and it leads to death, right? Cool little, super dark, yet interesting A&P side point. All right, so multiple cytochromes. This is my type of labeling, using the alphabet. I like to keeps it simple, right? Different species of bacteria have different cytochromes present. For example, the oxidase test detects the activity of cytochrome C oxidase. Now, right, some of your cells also have cytochrome C oxidase, but not all bacteria do. So you guys should have done an oxidase test. Does anyone remember the oxidase test? Dare I ask what color it turned? Okay, good. Purple, blue. Okay, you can take that. You're like, orange. Right, good. So cytochrome C oxidase, right, you had your little cassette, you were rubbing the bacteria on it, and it was detecting the presence of that enzyme, meaning that bacteria had cytochrome C oxidase. And so that's kind of nice because it tells you a couple things, right? It has a unique cytochrome. It also tells you that bacteria has an electron transport chain, because that's where you would find this carrier, right? So Neisseria, Pseudomonas, and Campylobacter are some of the big genre that have them, but there are other bacteria also. So it can be a pretty important distinguishing factor. Is Neisseria or Pseudomonas, do you guys remember, is that on your list? Pseudomonas is? Okay. All right, we're not going to talk too long about flavoproteins, but if you've taken vitamin B2, and then you're taking riboflavin, right? FAD is a flavin, right? So these guys, or girls, depending on, I guess, the sex you want to assign, to a molecule that doesn't have any, right, are great at carrying electrons and protons and can do this throughout the cytoplasmic membrane, or what other membrane, Alanis? Mondays. Gosh. Oh, that's so awesome. Okay, this is like this is gonna stick with me for a while. In a good way, right? A funny way. So it can carry electrons and protons through the cytoplasmic membrane, or what other membrane? In you? Where's the electron transport chain taking place in you? Yeah, I was about to fall over and just teach from laying down on the ground. But you got it. Yes. Thank you, Alanis. All right. So we don't have a ton of time here, but I just wanted to point out the electron transport chain in eukaryotic cells. And then you're going to see, hopefully today, but if not today, on Friday, some important key differences between the electron transport chain in eukaryotes and prokaryotic cells. So here are your different complexes, right? Notice this is taking place in the mitochondrial membrane, all right? You have NADH here dropping off protons to complex 1. For whatever reason, my old textbook, I guess, like to pretend that FADH2 didn't exist, but it would drop off its electrons here at complex 2. As Holmes pointed out, right, proton pump here, not a proton pump here. So FADH2 is not actually leading to protons being pumped out until complex 3. So again, you're not getting as much ATP per molecule there. All right. You then go to complex 4, which is another proton pump, and then ATP synthase, which again, maybe I'm crazy, but I used to remember it as complex 5. To each their own, right? Know that the proton gradient that's established here, right? These protons then rush in. When they rush in, it's very mechanical, right? You caused ATP synthase to spin, right? And as ATP synthase is spinning, right, is grabbing a phosphate and putting it onto ADP to give you ATP. This seems like common sense, right? Or you guys have heard, well, the proton gradient in your cells is used to make ATP. And that is, right? You guys have probably heard that since high school. But that's important to hold on to because you already know of one thing, and I think I've actually mentioned the other before previously. There's two other things that the proton gradient can be used for in the electron transport chain of prokaryotic cells. This is everything that I just said, right? So you can go back and read this yourself. What I do want to spend my last couple minutes on is this slide. Now, if you would put this slide next to the slide of the electron transport chain in eukaryotic cells, you would see those differences. I think even without me having those side by side, you can probably already notice that there's some differences. And so, right, one thing I want to point out is they don't have, right, as many complexes. In fact, prokaryotic cells are missing what we would call complex 3 in your cells, right? So they have one less proton pump. Okay. But their NADH dehydrogenase is the equivalent of your complex 1. The succinate dehydrogenase is the equivalent of your complex 2. And then ubiquinol oxidase is the equivalent of your complex 4. All right. But same general principle, FADH would drop off here. So again, they're not getting as much energy from FADH, just like you are not getting as much energy. And then here's one of the big differences, all right, is that not only can this proton gradient be used for ATP synthase, it can also be used for active transport, right? So you are basically coupling different molecules to these hydrogens, or not coupling, you don't actually attach them, but you're using, right, the fact that these hydrogen ions want to rush into your advantage to basically co-transport other molecules, which we do see kind of similar functions, although not from the proton gradient that's established in the mitochondria, but we do have similar mechanisms in eukaryotic cells. And then to power your flagella, remember we talked about that, the proton gradient powering the flagella. So your proton gradient you're using for one main function. Their proton gradient can be used for potentially 3 different functions. Right? And we'll point out here in just a little bit that this is also an important difference, right? We do have in some cases, and I'm going to end here, in some cases, they can actually use different inorganic molecules as their terminal electron acceptor, meaning in the absence of oxygen, they are still able to carry out electron transport. And that's really, really advantageous, right? In the absence of oxygen, they still can produce a large amount of energy. If I take oxygen away from your cells, which they can, right? That can happen for a little while, right? If any of you have seen me and Michael in the gym getting swole, right? Pumping iron, right? Again, when you have strenuous exercise over a long period of time, right, you start to use the oxygen stores up in your cells, and then your cells, right, start to undergo, right, fermentation to generate energy, right? Lactic acid's produced, just like Jennifer Buckner said, and that's one of the reasons that after a good workout, you're really sore, is because you have acid that's accumulated in those muscles. All right, cool. Oh, yeah, so this is where we're gonna start on Friday. I will see you guys then. I will email you back again after my gateway class, so it's got a little while, but we'll try to get something